Perfect. Thank you so
Perfect.
much. Thank you so much for the kind introduction, and thank you so much for letting me show you our latest data. So I'm gonna talk to you about sensory processing dysregulation and using that one appling insufficiency. This is the work that I've done for most part of my postdoc in Dr. De Cristo's lab here in Montreal. And so first, what is SYNGAP1? Most of you might not know it probably because it's a neurodevelopmental disorder that it has been very recently described, actually only on 29. It was described as a neurodevelopmental disorder by a group here in San Luis, where I work. And this is an epileptic encephalopathy that is characterized by severe intellectual disability. It's also comorbid with autism spectrum disorder. They have sleep disorder. So it's very recent. We know little, and to date there more than 700 individuals diagnosed with SYNGAP1 related disorder, this number is increasing super quickly thanks to the genetic testing that it's been developed very recently as well. So yeah, as I said, we know very little, and it's been recently described, so no effective drug has been developed to help these people. So of course, one of our urgencies was to find a proper drug, a drug that could at least help these patients. But of course, since we didn't know anything, we know very little in the literature. There's very little about what are the underlying mechanism or even sensory perception. It's been very little investigated so until now. So we were wondering whether the first thing to do before starting trying drugs like crazy, it was find robust biomarkers that will work in the same way in humans than in mice. We've known that there are many, unfortunately, there are many studies that have developed interesting drug treatment that work very well in mice, but as soon as we move this to clinical trial, they fail. So one of our priorities before starting any drug screening was just that, trying to find these biomarkers. And the biomarkers that we are using, they are electrophysiological biomarker. In children, in patients, we just record electrophysiological activity from the brain in a non-invasive way, just using this mesh that you can see here. So it's non-invasive, no pain or anything. And in mice, since we had the chance to go intracranial, we also so we just perform a surgery where we introduce electrodes in 3 different areas because I told you that we wanted to explore sensory perception, in particular auditory and visual perception. So first thing that I wanted to show you here, even if I told you already that epileptic activity was one of the main symptoms in this patient, is that since we are looking for fast and robust biomarker for this disorder, I just we just proposed this intellectual sites as a very fast and robust biomarker, because most of the time when people work in epilepsy, we have to do very long recording in order to catch this seizure, epileptic seizure, because they can appear some sparsely sometimes. However, here we show that these interictal spikes, only with 10 minutes of recording, we can already see this huge phenotype. So this is for interictal spikes. I'm gonna rush a little bit because, okay, so then we go to the resting state. We just wanted to look at what it was the oscillatory activity in the resting state in both mice and patients. And we observed that we observed a huge increase in the gamma band, in the whole gamma band in both cases. So we were happy with this similarity between mice and patients. And then we went to look at the sensory perception in this patient and this mice model. So first we start by looking at just a sequence of image and sounds that are randomly presented, so they cannot be predicted. And when it comes to and then we perform just a grand average analysis, which is the classical analysis that is done when people look at evoked related potential. It was good for the visual response because it gave us similar response in both mice and humans. In both cases, we obtained a huge decrease in the response. But when it comes to the auditory response, we observe like the opposite. Higher hypersensitivity in mice, while it was very small response in patients. So these analyses were okay, and data, but we wanted to go forward and look at other analyses that they might tell us a little bit more about what is going on during this simulation. So these analyses, a little bit more complicated, are phase amplitude coupling. And just to tell you a little bit what it is, it's okay, when we record electrolytical activity from the brain, we have kind of a mix of activities of all the neurons that are surrounding our electrodes. And this mixed activity, it contains a huge amount of different frequencies that are embedded all happening at the same time. So when you do the filtering of your raw signal, you can when you filter your raw signal at different bands, you can see the prominence of each different band region, brain region. So brain phase amplitude coupling is basically the phenomenon by which low frequency, so the phase of the low frequency modulate the amplitude of the high frequency. In that way, so this is supposed to be a resource of the brain for different networks to communicate faster and more efficiently. It's been shown to increase this coupling. It's been shown to increase during decision making and so on. So we were expecting that in our patients and animals, we will have less phase amplitude coupling, but the opposite. We observed higher phase amplitude coupling. This it was surprising at the beginning, but then we started looking at the bibliography, and we observed that actually it's not that strange. This increase in phase amplitude coupling has also been described in the motor cortex of Parkinsonian patients during repetitive movement, and it's also been described in epileptic patients. So we observed this increase in face-up-repeat coupling in both mouse and human, in both auditory here and doing both auditory and visual stimulation. So then we went to again simple passive sensory responses that a little bit more complicated, that are auditory habituation and deviant detection. So quickly, habituation is just the mechanism by which the brain is able to reduce the response to a repetitive stimuli. So like this, we can end up by ignoring repetitive and non-important stimuli that are repeating themselves. Like, for example, when we start studying in the morning and there is a machine beside us, after 10 minutes, we don't hear it anymore. So we can ignore this unuseful information to concentrate, actually to be able to do the deviant detection, which is the opposite mechanism by which the brain is able to highlight the novel information that it's interesting. So we studied these two functions in our patients and mice, and we observed. So this is the typical response after one. Again, these are grand average analysis, the classical one. So when we stimulate with the first simulation produced a normal, a big response, but for the second already you can see an important decrease in the amplitude. So this decrease in the amplitude is what we call habituation. Habituation, we can see that it doesn't happen in our SYNGAP mice or patients. Here is just the complications. So we observe this lack of habituation in SYNGAP subjects. When we look at the deviant detection, so just here you can see the response to the standard sound and the response to the deviant. The way it's usually studied the deviant detection is by mismatch negativity, which is just the subtraction of the deviant response minus the standard response of the brain. And this is the typical pattern that we observe when we do it in control mice or humans. The blue one are control. And when we superpose the response, you can see that the SYNGAP mice, in this case, this is mouse data, we observe abnormal response at the P1 component, which is the most important and critical for mismatch negativity. So yeah, just to summarize the first part, we observed these different biomarkers that we aim to use for drug screening, and we hope that it will also help other labs for their drug screening. But we also wonder, we mainly wonder, what are the neural circuits underlying these abnormalities? Why is it happening? And to answer this question, we use a genetic approach in which we remove SYNGAP, one copy of SYNGAP from MG-derived neurons, which are mainly PD and somatostatin cells, interneuron cell, inhibitory cells, and this selective removal of this gene gave us, surprisingly, so at the beginning I showed you that the increase of gamma it was in all the different brain regions that we explore. However, here the increase in gamma is selected to the auditory cortex, and a little bit of high gamma in the visual cortex for this conditional heterozygotes. And when we analyze the evoked response of during a pure tone presentation, we observe again a difference in the P1 component, but not in the A1. So this hyper excitability that we observed in the germline mouse, we didn't get it in the conditional heterozygotes. When we do the power spectrum density, we observe an increased evoked gamma during pure tone, as well as an increased phase amplitude coupling, as we observed in the germline, suggesting that in this biomarker, PV and somatostatin interneurons might be very much implicated. However, when we go to the visual, as you could expect it from the first, from the power spectrum density during resting state, during visual stimulation, we didn't get much difference. We couldn't replicate. I mean, it doesn't show the same results that we observed in the germline. Only just a little bit of gamma, evoked gamma increased during visual stimulation. That's all we didn't get the power spectrum, the phase amplitude coupling. And when we go to the adaptation to repetitive sounds, the one that I showed you a few minutes ago, we observe again that in our control, there is an habituation that we don't observe in heterozygote mice, conditional heterozygote mice. And the deviant detection, it's unaltered. All these results just show us that, okay, first we identify sensory response abnormalities that can be robust and can be used as robust and translational biomarkers for SYNGAP1 abnormal. But most interestingly for me, it's also that GABAergic circuits play an important role in the generation of some of those sensory abnormalities, but not others. And especially highlighting the difference between auditory and visual modalities. That's something that intrigues me, and something that we are investigating right now. Just I want to thank Graciela's lab. This is only a little part of a huge project, and yeah, especially thanking Vidia, Ruggiero, and Virisha for their help and their work. And funding agencies and families, their cooperation help us and make possible all this research. So yeah, thank you so much. Yes, let's ask as many questions as you want, because I think I went a little bit fast.